President Randy, and good afternoon, everybody. A quick announcement before I introduce our speaker. Sadly, our program next week, which was going to be a recording from the Kent Roosevelt Aces, we're going to have to change that because school has been changing. But I promise I've got something very interesting and fun in store, so please look forward to that next Tuesday. Today, our speaker is Dr. Rita Caldwell. Dr. Caldwell was born in Beverly, Massachusetts, and holds a Bachelor of Science in Bacteriology and a Master of Science in Genetics from Purdue University. She earned her PhD in Oceanography from the University of Washington. Her interests are focused on global infectious diseases, water, education, and health. Dr. Caldwell served as the 11th Director of the National Science Foundation from 1998 to 2004. In her capacity as NSF Director, she served as co-chair of the Committee on Science of the National Science and Technology Council. One of her major interests includes K-12 science and mathematics education, graduate science and engineering education, and increased participation of women and minorities in science and engineering. Dr. Caldwell has held many advisory positions in the U.S. government, nonprofit science policy organizations, and private foundations, as well as the international scientific research community. She is a nationally respected scientist and educator, has authored or co-authored 19 books and more than 800 scientific publications. She produced the award-winning film Invisible Seas and has served on editorial boards of numerous scientific journals. She serves as chair of the research board for the Gulf of Mexico Research Initiative. Her latest book is A Lab of One's Own, One Woman's Personal Journey Through Sexism in Science. She has been recognized around the globe with the highest science honors. If I read the complete list, I believe I would become today's program, but to list just a few, she has been awarded 63 honorary degrees from institutions of higher education, received the 2006 National Medal of Science awarded by the President of the United States, and has received awards from the Emperor of Japan, the King of Sweden, and many, many other dignitaries. Maybe most interesting of all is that a geological site in Antarctica, the Caldwell Massif, has been named in recognition of her work in the polar regions. I'm not sure if one person's background could cover Rotary's 7 areas of focus any better than Dr. Caldwell's. We're so pleased to have her with us today from Bethesda, Maryland, Dr. Rita Caldwell.
Thank you very much, Dave. I really appreciate the kind introduction, and I hope that I'm going to be able to get the slides up. I had them up neatly just a bit ago. Let's see, I need to share screen. Well, it's going to be a bit of a problem here. So let me try something else. I think I should have left the slides up again. What I'm going to talk about today is the work I've done on cholera, and this goes back about 30 or 40 years, and how it has fit into what we're now working on COVID, and how I'm able to be able to use the data that we've gathered. Let's see if this will work. Okay. I think I should have left the slides up, taking a bit. Okay. Now to share the slides. Let's see. I wonder if you can show the slides from there. Well, wait a minute. I might here. I guess you're going to have to show them from there for some reason.
You bring those up, Dave?
Yeah, I'm having some problem. I'm not able to.
I'll be able to bring them up in just 1 second.
Yeah, that would be terrific. I had them all set, but they've gone into the ether. work started in the Chesapeake Bay and moved on to Bangladesh and India, and it's been very interesting that good, that's great. So if you can go to the next slide, yeah, that would be good. The important point that I want to make here is, and this is something I know the Rotary Club has contributed to in the past, and that is providing safe water in various parts of the world. The diseases that are transmitted by water are about 20 or 30 of them, and cholera is the one that's most dominant because it's about 1.5 billion cases every year, and at least anywhere from 1.5 to 2 million deaths. We are, may I have the next slide? We really are in a pandemic already. We're in two pandemics, the COVID, which we all know about, but we're also in the cholera pandemic because it started in the 1960s and it occurs every year in about 50 countries. It affects about 7 million people. And the Bengal Delta, you know, the coast just off Bangladesh in the Bay of Bengal is we think today the home of cholera, but you know, 100 years ago, there was cholera in Washington, D.C., in New York, and in Savannah, Georgia, in Canada, in Europe. And one of the discoveries, we have the next slide, one of the discoveries that I made with my team about 40 years ago, when I started my work, working in Chesapeake Bay, those numbers are the sampling stations that we started sampling back then, and we continue to sample. I myself don't do it now, my students do, and my students are doing this. But the water samples were collected, next slide. And one of the discoveries was that the bacteria that cause cholera are naturally occurring in the environment. They're part of the natural flora, and they're associated with plankton, with zooplankton. Now you can see the cholera bacterium, that's the curve-shaped rod with a little tail that gives it motility. It's associated with these plankton copepods. Now the next slide. The copepods shown here, this is a female, and you can see the egg sac on the left. The bacteria coat the surface of the egg sac. They have a very powerful enzyme which causes the sac to break. And I think it's a relationship that's probably symbiotic. Maybe the next slide. So we developed this model of cholera back, I say about 1970, 75, where when in the spring, when there's a lot of sun, the chlorophyll-bearing plankton become very populated, they bloom, they become abundant. In fact, we now know that there are algal blooms that are not so good for us, but the phytoplankton then are food for the zooplankton. They become dominant, and then when the food source declines and lessens, then the plankton break up, and then the bacteria are released into the water column. And when there's no treatment of the water, then you can have the disease transmitted. Now, this was a hypothesis we developed in the US, in the Chesapeake Bay, and we were criticized that, well, you know, does that really work in Bangladesh? So next slide. We went to Bangladesh. We went to Dhaka and then to the remote villages right on the coast of Bangladesh and India, near Calcutta. Now the next slide. And indeed, the source of water for the villagers is these ponds. The ponds are polluted, but that's all the water they have. The ponds serve not just as a drinking source, but also where they wash the dishes, where they wash the food for the meals, where they brush their teeth, etc. So clearly not a terribly sanitary situation. Now the next slide. And so we found out that yes, the same paradigm that we discovered in Chesapeake Bay exists in Bangladesh and India. Of course, there's person-to-person transmission there because of the poor sanitary conditions, but fundamentally the bacteria are from the environment. They're in the environment and they play a role in the nitrogen and carbon cycling. We have the next slide. And so it occurred to me back in 1985 when Landsat, the satellite, was launched. And Landsat had sensors that detected chlorophyll and measured sea surface temperature and sea surface height. And all of these factors play a role in cholera. So my idea was, well, maybe we could predict cholera by satellite. So sure enough, next slide. What we did was show that that blue line is the sea surface temperature in a 1 mile quadrant just offshore in the Bay of Bengal. And the red line are the cholera cases that occur in that region in Bangladesh. In other words, we got a really rather good fit when you're measuring sea surface temperature, chlorophyll, and you're matching it with a pretty good correlation with the number of cases of cholera. We have the next slide. So that we looked into this in great detail. A colleague of mine um, um, from New Zealand, a physician, had done a PhD in India on water systems, and then her medical degree, and she was practicing in London. And Dr. Elizabeth Whitcomb discovered the records for cholera, malaria, meticulously kept by the British colonials in India. Every death from cholera, of course, they called it diarrhea, and every death from malaria, they called it I think it was the sickness or whatever. But they recorded it on a daily basis. And also, the British colonials recorded the meteorological data, rainfall, temperature, etc. So we put all this together. Next slide. And we discovered that there's a pattern in cholera. Along the coast, there's cholera all year long, with a big peak in the spring and again in the fall, which matches the plankton blooms. But inland around Delhi, near the river, there would be epidemics. You know, suddenly you'd have a massive um, thousands of cases, and then 30, 40 years, no cholera at all. And so next slide, we perfected our model based on the fact that when the temperature for the preceding 2 or 3 weeks was very high and began to increase, the number of cholera cases increased. So there was a direct relationship between the temperature, the air temperature, and rainfall. Next slide. And so other factors like salinity, dissolved organic, a variety of factors began to accumulate. When we started publishing our papers, other investigators got interested in the environmental aspects of this massive pandemic causing agent, Vibrio cholerae. We have the next slide, please. We improved our model. It's now quite sophisticated. We include the simple factors like temperature, rainfall, lack of good sanitation, but also other factors like salinity and nutrients and so forth. Next slide. And we're using a much more sophisticated set of satellite sensors. The first, of course, was back in 1996 when we did our study with the Landsat. But now in 2015 and 16 and 18, we're using satellites that measure density of populations, migration of populations, humidity. It's quite amazing, all of the factors that we can now build into our model. Next slide, please. And so we asked ourselves, since this model was working so well in Bangladesh, could we have predicted the cholera outbreak in Haiti? As you may remember, in 2010, and I suspect some Rotarians were helping out in Haiti as well, but in 2010, there was a massive earthquake. It was a force 10. It was really a very severe earthquake. And it was followed in the fall, around October, November, with the worst cholera epidemic at that time that had been recorded historically. We have the next slide. And so what we determined that the location of the original contamination was inland, and it was apparently and theoretically ascribed to the UN peacekeepers. However, at the same time as the presumed initial outbreak, all these other cases along the coast were occurring. So it struck us and me as really an environmental factor. Now, they said at the time that there had never been cholera in Haiti. Well, a couple of years ago, well after the earthquake and the big outbreak in 2010, in 2008, a scientist discovered samples of stool of a study that had been done of infants with severe diarrhea in Haiti. And those samples collected in 2008, 2 years before the massive cholera outbreak, they detected the presence of Vibrio cholerae, the causative agent of cholera. So the bacterium has been in the environment before the earthquake. Have the next slide, please. And so what we discovered was, going into the literature and into the archives, that in 2010, the air temperature was the hottest in 50 years. That's the red line. And the rainfall that September, October, because of a massive hurricane that roared through Haiti, it was the heaviest rainfall in 50 years. This fit the pattern that we had discovered from the studies of India from 1823 to 1930 or 1940, those data that we had converted from the books in the maps in the British Museum to the computer. Yeah, the next slide. And so we looked at Hurricane Matthew, which spread along the coast, across Haiti and up along the US coast. And we from the data, we did a prediction using our model. Next slide. And our prediction is on the left, and the actual cases you can see on the right. On the left, you can see the very dark red area is where we predicted the highest risk of cholera in that country. And you can see that our prediction really was borne out. Now this intrigued us, because then there was another massive outbreak of cholera in Yemen. And so we took the same model, satellite imagery and prediction. Next slide. And we did a retrospective study of in 2017. And you can see on the left, the blue is very low risk, the red is very high risk, and you can see the lower right where the actual cases were occurring. And again, our prediction was borne out. We published the paper quickly. It was picked up by Scientific American, and then we got a phone call in January of 2018 from a colleague at the British Aid Agency in England. He said, look, I saw the Scientific American article. I then went to your paper, and we would like to work with you. If you would predict for us where the highest risk is, we'll position physicians, medical supplies, and safe water sources in those regions. So that's what we did in 2018. And in fact, the cholera rate went down significantly in 2018. Next slide. So we've been working ever since with the British Aid Agency to provide them every 2 weeks. We provide them a 4-week prospective risk analysis, so they can be prepared as to when and where the outbreaks are most likely to occur. And we've been working with them through 2018, 19, and we continue to work with them. In addition, UNICEF and the British Meteorological Agency, along with NASA, which provides the funding for us, we've been all working together on this um, predictive capacity, risk capacity. May I have the next slide, please? Now, let me point out that cholera is the bacteria are susceptible to global warming. And let me explain that. Shown here, those red squares are sampling sites. What happened was the British Marine Station, Southampton, decided about 40 years ago that they would start archiving plankton samples. So they sent out these plankton nets to yachtsmen, shipping captains, to cruising boat captains, to ferry boat captains, and said, look, collect a plankton sample. They showed them how to do it. Collect a sample of plankton, send the sample to us, and give us the coordinates, the longitude and latitude. And so all of these samples are in the British Marine Station. And so my colleagues in Italy, Dr. Carla Pruzzo, and my other colleagues in Germany at the Max Planck Institute, and the Southampton scientists and us at Maryland got together. And what we did was to go to those samples of plankton, extract the DNA, and then probe it with specific probes for the cholera vibrio and other pathogenic vibrios. We have the next slide. So you can see that the red line is the sea surface temperature, and the triangles are the increased numbers of bacteria. May I have the next slide, please? And so the interesting thing was we could show together this team of Germans, Italians, and British Americans working together, we could show that as the temperature of the water in the North Atlantic increased, the numbers of bacteria became more abundant and the infections increased. And on the bottom is along the US coast from Massachusetts, Maine, and up in Canada with Vibrio parahemolyticus, which is a food pathogen. And the top shows the increase in the numbers of occasional cases of cholera, Vibrio parahemolyticus, and also a very bad bacterium, Vibrio vulnificus. And so you can see this is the first correlation of global warming of the ocean temperature and an infectious disease in humans. We have the next slide, please. I'm going to, I wonder if whoever it is, could go on mute. Somebody could go on mute, that would be great. Anyway, the other technique that we use in our studies is DNA sequencing that I've just described. May I have the next slide? We developed a method that allows us to extract DNA, sequence it in machines, and then match up the sequence bits with a database that gives us great identification, as well as being able to determine the presence of antibiotic resistance genes, etc. May I have the next slide, please? And so we can identify right down to strain. Now, this is important because if you're manufacturing cheese, you use one strain of Lactobacillus casei. If you're manufacturing Chardonnay, you use another strain of Lactobacillus casei. But if you do use a third strain, you may be making yogurt. We have the next slide. So we were able to, and this is a good example of why it's important to identify strain. Here's a young woman who built a zip line. The zip line broke. She developed a severe gash because she landed in the water on the rocks and ended up with a serious infection of necrotizing cellular fasciitis. The disease unfortunately progressed so that she had to have her legs amputated to the knees and her arms to the elbow. She did survive. And then water bacteria, 3 strains were isolated. 3 of them proved to be identical and the others seemed to be different. So we injected all 4 strains into mice to see how this disease progressed. And it was very interesting because, next slide, only 1 strain went to the liver and spleen, in other words, went systemic. And to make a long story short, next slide, we confirmed this by making that 1 strain bioluminescent and kanamycin resistant. And sure enough, that was the only strain that went to the liver and spleen. And so, to make the long story short, these bacteria worked together to the 1 set of strains broke down the muscle tissue, and that allowed the other strain to go to the spleen and the liver. So this is one of the strong techniques that we've developed. Next slide, please. I'll go through this rather quickly. Next slide. We've studied the gut flora, that is in a hospital in Calcutta, in 3 tranches, phase 1, 2, and 3. They sent us samples. In the first tranche of samples, they could isolate and detect Vibrio cholerae. In the next phase, they couldn't, but the patients still had severe disease, just like cholera. And then the third phase, the additional samples. May I have the next slide? And so this, it looks complicated, but it really isn't. In the top left, these are the diarrheal patients where they could isolate Vibrio cholerae. We could, but we also isolated other pathogens. So we begin to think that cholera is really a mixture, not only of Vibrio cholerae, but other bacteria. But where they could not identify Vibrio cholerae or the cholera bacterium, the top right, we were able to show that it was a mixture of enterobacteria pathogens. And then the bottom left, interestingly, those were the healthy population, and the pink area is the pathogens. And then in the bottom right, this is Westerners, the NIH study on gut flora of us Americans. And as you can see, there's a pattern here. Next slide. That pattern is very, let me skip that. Next slide. The pattern is such that not only can we detect antibiotic resistance, of which there's a tremendous amount in India because they can get antibiotics without a prescription and even from street peddlers. May I have the next slide, please? We were able to put all the data together to get kind of the normal flora of globally of samples from 10 countries, 600 samples. Next slide. But the important thing is that the samples then can be used. For some reason, this doesn't show very well, but oh, okay, it's a little slow. The samples from different countries provide essentially a forensic tool. And I don't think I'll wait for this whole thing to show, but fundamentally what it shows is that for each country, in this case, US and other countries, by the time this shows up, it'll be a bit slow. In any case, it's a good forensic tool. May I have the next slide? Let's go on to the next one. It's a public health tool because we're able to identify the bacteria, the fungus, the viruses, and the parasites. And this is a great tool because it can map what's occurring in different countries around the world. May I have the next slide, please? We're looking at doing a lot of work with hospitals. In this case, we're doing a big study with Harvard Medical School looking at celiac disease. May I have the next slide? And we're able to show differences in those who are exposed to antibiotics and not exposed to antibiotics, those who have the celiac disease genes and those that do not. May I have the next slide? Crohn's disease is a little more dramatic because there we are looking at male and female adults. May I have the next slide? And it's very clear, the green are the bacteria in healthy individuals and the red are the bacteria in the Crohn's disease individuals. So this becomes a diagnostic. May I have the next slide, please? So let me just give you some practical examples of this DNA technique so that you can understand why I'm showing it to you. And that is, next slide. We've been doing a big study with Orange County California. That's the Los Angeles area. You know, of course, that California has had a severe drought, and they've had a rather difficult problem getting enough water into the drinking water plant from the groundwater in the river and the lakes. And so they're reusing the water from the sewage plant. Question is that safe? So the water comes in, Q1, that's the water that comes in from the sewage plant to the drinking water plant. It's been screened, and then it goes through microfiltration, reverse osmosis, UV irradiation, and then a whole series of other steps of chlorination and other kinds of treatment. Have the next slide. So when we looked at the sample of water, that gray area, that circle area, all kinds of bacteria, including pathogens. But by the time it goes through reverse osmosis and microfiltration, you can see to the left, the little blue dots, those are water bacteria, harmless, and in any tap water that we would be testing. Have the next slide. And for the viruses, the same thing. Lots of viruses, including adenovirus and other human viruses in the Q1 coming from the sewage plant. But by the time it goes through microfiltration and reverse osmosis, hardly any viruses, and they're mostly bacterial viruses, which aren't pathogenic for humans. And by further treatment, almost zero viruses, just a few of the bacterial viruses in the drinking water supply. May I have the next slide, please? And there was a concern that with the sewage bacteria going into the drinking water plant, they would transfer the antibiotic resistance to the naturally occurring water bacteria. But that's not the case, because we could determine that. We could show that the antibiotic resistance transfer does not occur. We have the next slide, please. What has all this got to do with COVID? Well, we now know, we've learned a heck of a lot in 10 months or 11 months since we had the first case in the US. We know that the virus, now I've been talking about cholera, that's a bacterium. Now I'm talking about a virus, but the behavior is quite similar. In the case of the COVID virus, though, it infects the lungs, but also the liver and the kidneys and the heart and blood vessels, even the brain. We know because one of the symptoms of severe cases is to lose the sense of smell and the sense of taste. Why does it affect all the systems? Because the receptor site, the ACE2 receptor, is common in humans on the various organs. And that is the explanation for all of the various components of the body being affected. We have the next slide, please. We are able, by that DNA technique and the RNA DNA, that is nucleic acid technique that I just explained, we tested, and we could, in a stool sample, we can isolate, and we don't have to isolate, we can detect directly the SARS-CoV virus that causes COVID-19 and its variants. So this DNA tool is very important. Next slide. It's been shown by others that in about 50% of the stool samples from different patients, you can detect virus. The virus is present in stool in victims. Next slide. Now this slide looks complicated. It's not. If you just read down from the top to the bottom, those are patients. And if you read across, the red line means that that patient throat swab, the standard COVID test, was positive. Notice how the orange line, the yellowish orange line, stays for quite a long time. That is, the stool shows the presence of the virus. So after the throat swab test has gone negative, you're still shedding virus. Next slide. This is important because that means we can detect the virus in sewage. And that's a mechanism, first of all, for early warning, because when the virus starts appearing in the stool sample, cases will then 4 to 7 days later will start appearing in the community. And conversely, when we've been testing and finding lots of virus, but then the concentration of virus goes down with each subsequent sample, that's an indication that the distancing, masking, hand washing is working, and that we can ease up a bit on the restrictions. So we've been doing this study for the last several months, and we will be for the next 3 years, for the state of Maryland, working with the Department of Environment, the Department of Health, and the governor's office. Next slide. Here's a typical sample of an analysis. You see the blue line, which is the set of genes that are used to determine the presence of the virus according to the Centers for Disease Control. The yellow-orange line is that we use additional viruses to make genes to make sure that we're really detecting the genes of the virus. The blue line genes, the end target genes of CDC, and then the genes that we, in addition, use to make sure that, in fact, the bottom chart then is an accurate chart that we can send to the governor. This was for Frederick, Maryland. And notice the big peak in July, July 21, 23, July 30. That, in fact, was an indication that in the community, there were cases that were asymptomatic, but there would soon be symptomatic cases, and indeed there were. And so now we've been providing every week, 4 or 5 times a week, we've been providing for different areas in the state of Maryland these data. We have the next slide. Now, what's very interesting, and what I'm very excited about, is all that work on satellite sensing of the risk factors for cholera. We've applied for counties of the United States risk factors for COVID-19. And shown on the left is our first risk map, which we produced in April, May. And then on the right are the actual cases at that time in the United States. We've improved our model for COVID with more of the COVID relevant information like humidity, dew point temperatures, cell phone data for movement of the population, et cetera, and for tracking cases. And so we now have then this satellite sensing prediction capability for COVID-19, which we're in the process of publishing. May I have the next slide, please? A little bit of data, just for the fun of it, about the metagenomics, that is the bacteria in drinking water. Next slide. What we did was collect bottled water from the supermarket, artesian well water, and then tap water from drinking fountains, just to see what's there. Next slide. And it turns out that you can just discriminate based on the kinds of bacteria that are present. The circled area with the green dots, that's the non-mineral bottled water from the supermarket. The blue dot at the top, that's sparkling mineral water that came from a natural source. And then in the bottom left, the star and the triangle were tap water. Next slide. So we can tell that predominantly just ordinary bacteria that you would find in any river or stream that are not pathogenic for humans. No fecal indicator bacteria, no E. coli, which is good, in none of the samples we tested. The only viruses we picked up were bacteria viruses, which we already knew from the work with the drinking water plant that we would expect to find that. And then the unusual DNA in mineral water was non-pathogenic, but just some ancient kinds of bacteria. So this is kind of interesting. Have the next slide, please. Let me close. And I think this is something that goes to the heart of Rotarians, helping people. All of the work that I've been describing for you, satellites, computational modeling, DNA analysis, We still have villagers in the remote parts of Bangladesh who have to drink water from the river, from ponds. How could we help them? Well, the hypothesis was if the cholera bacteria are associated with plankton, plankton are kind of the elephants of the microscopic world. If we could filter out the plankton and the particulate matter, we could reduce cholera. Because from the laboratory studies we did, we tested it. We tried men's t-shirt material, ladies' sari cloth material, and even some expensive Chinese poplin material, which which is available in Bangladesh, but it's very expensive. Only the rich can afford it. Well, in our lab studies, we found that we could filter plankton using sari cloth. Next slide. Folded about 4 or 5 times. And so in a study done that was funded by the NIH, the Nursing Institute, it was a 3-year study in about 100 villages and thousands of villagers. We used women as extension agents. We trained them, we employed them to go out to the remote villages and show how to filter the water and make it safe for their families and their babies. Next slide. It was important to show them that they should take about a yard of old sari cloth, used, cheap, inexpensive. Everybody had it, didn't have to pay for it. Folded it 4 or 5 times, put it over the mouth of the calash, collect the water by filtering, and then at the end, unfold the sari cloth, rinse it, and dry it in sunlight because that acts as a disinfectant. Sunlight is a good disinfectant. May I have the next slide? It wasn't difficult to explain how important this was because to the right is filtered water, to the left is water with things swimming in it, and explaining that's not good for children, for babies. It's a very, very quick learning lesson. May I have the next slide, please? We were able to reduce cholera 50%, just about 50% in these remote villages just by this simple technique. But it's evidence-based, it's science-based, but it's simplistic and it's applicable in a remote area to provide a benefit based on all the work that we have done to the remote villagers in Bangladesh. May I have the next slide? Lots of people I have to acknowledge, and I'm delighted to acknowledge, from the International Center for Diarrheal Diseases Research in Bangladesh, from the National Cholera Institute in Calcutta, India. Wonderful students. Kyle Brumfield is the most recent student who did the bottled water study that I just showed, data I just showed, but many other postdocs, graduate students. Next slide. And colleagues from NASA, from other countries, from other agencies. Next slide, please. I want to do a shout out for Dr. Huck. He and I have worked together for 40 years since I first met him in Bangladesh, talked him into becoming a graduate student, and he's now a research professor at the University of Maryland, and we work together. Dr. Antar Jutla, a postdoc who is now an associate professor at the University of Florida in the College of Engineering. He's an engineer, and he's responsible for the latest elegant work on satellite imaging. Dr. Hassan, who worked with me in the company that I founded, Cosmos ID, to do the DNA sequencing and medical work. He now has founded his own company. And then Dr. Sion Young Choi, who is responsible for the massive database, and it's highly curated, very clean and great condition. She is a wonderful scientist. May I have the next slide, please? And then I really want to shout out to the Rotarians. Indeed, safe water is a global challenge, and thank you for the programs that you have funded in the past and are continuing to fund. Next slide. A lot of this is written up in the book that I published in August. It tells a story of other interesting tales, including the anthrax story. I chaired the anthrax committee that aided the FBI and the CIA in 2001, 2002, now after 9/11, to track down the perpetrator. Next slide. glad to take any questions, and a pleasure and a delight to speak with you, and look forward to any discussion or questions. Thank you. Thank you, Ken, for showing the slides. As they say in Brooklyn, you done good.
Dr. Caldwell, do you have can you tell if there's a threshold of temperature, a temperature threshold where the cholera starts to generate?
Yes, it's about 15, well, the salinity is about 15 parts per thousand, and the temperature is about 20 between 20 and 25 C. And then when it gets to be 28 or 30, then it's real probability goes way up. So that would be Fahrenheit. That would be around 90, 95 when it really gets to be a serious problem. When it's around 70 is when 75 or 80 is when cases begin to increase in India.
Thank you very much.
I think, Dr. Caldwell, we have a custom in our club that someone responds. And I think we'd like to do that right now, but can you hang around for a few minutes afterwards in case there are other questions?
Sure, be delighted to, Ken.
That would be great. Okay, Kurt, go ahead with your response.
Well, Dr. Caldwell, that was an extremely informative and diverse presentation. Thank you so much for all the details on the different projects you've worked on. I'm sure that you could have spoken for several more hours about each one of those projects, and I can tell how much passion and dedication you have in your presentation. You know, I always try to pick out a few things that stand out from a speaker, and I think the things that stood out from your presentation were using science as a method to predict, model, and solve problems. And I think that's what everyone, as a scientist myself, and as most people in science, that's their goal, is to study something and utilize that knowledge to help the world become a better place and to solve problems. And I think you gave a great explanation about cholera and using the predictive modeling that you had to set up preparations for places that might have increases in cholera. Water reuse, using water and actually being able to reuse that in places where there's a lack of water. Showing science, using science to prove that it is a safe process. So again, I think if there was one take-home message, I think in this world we live in now, where science sometimes is put into question and is sometimes thrown to the gutter because of pundits and because of discussions, I think we all need to understand how important science has been in the past and is in the future for solving problems. I think you gave some great explanations for that. So thank you very much for your time. I'm so glad that Dave was listening to NPR and heard you and contacted you and was able to get you to talk to us. And I'm sure there's going to be several more good questions here, so I don't want to ramble too long. But again, thank you so much.
Thank you. Very well spent, and I really agree with you totally. Very well said.
Thank you much, Kurt, and thank you, Dr. Caldwell. We're going to officially end the meeting, but as I say, if anyone has questions, please hang around.
Dr. Caldwell, were you at the University of Washington when my friend Jim Purdue